So in the second podcast on integument, we're going to finish just a little brief part about the deeper layers of the skin, but then we're going to move into hair and the nails. So in the deeper layers, especially the reticular layers of the skin, we're going to be looking at sweat glands, sebaceous glands. Certainly then we're going to be looking at the hair and the hair follicles and all the different layers associated with the hair. And actually we're going to see it. Hair is simply an invagination of our epidermal layer and modification, of course. And then we'll look at the different layers of our nails. So we left the papillary layers. We're moving deeper in the dermis, and the reticular layer continues to be dense irregular connective tissue and much more coarse fibers that we see. Very thick elastic fibers, type 1 collagen, those reticular layers. And we're going to find the same cells that we have there, but then again, as we get closer to the hypodermal layer, we will also begin to see adipose tissue occurring much more. Now, sweat glands, sebaceous glands, hair follicles, these are going to be flowing through the dermis, especially in thin skin. Remember, in thick skin, we will see the sweat glands, but not the sebaceous and the hair follicles. Now, we'll also see little bundles of smooth muscle attached to the hair follicles, and these are going to be the erector pili muscles, and those help raise the hair in moments where you're cold, where you're frightened. And here in this particular photomicrograph, we can see our hair follicle, and then we have this little band of smooth muscle attaching to the hair follicle and the other tissues. So when this contracts, it's going to pull this hair follicle in this direction, almost like standing up a flagpole. And as you do that, the skin on this side of the hair follicle is going to raise up, and that's going to be the goosebump or the chill bump that you see when that happens. Now, also in these layers, we have other receptors. The one that's most recognizable in this layer is going to be the Pacinian corpuscle. This is going to essentially look like an onion, a lot of concentric rings, encapsulated structure. And this is going to be mechanoreceptor against pressure and vibration. Then we also have the Ruffini corpuscles. I learned them as Ruffinian, but they're Ruffini corpuscles now. We won't identify these, but these are going to be involved in the stretch of the collagen fibers present in the reticular layer. Now, in this micrograph, we can see some of the deeper layers of our skin. We can see a lot of our... This is going to be in the deeper layers of our skin, and we can recognize our reticular fibers. Here, we've got a blood vessel collapsed here, likely a vein. Here we see some of the ducts, the darker regions, some of the acini of our sweat glands here, and we'll look at those more in a little bit down the road here, a few more slides. We got some adipose tissue, and then we see this onion-like concentric ring structure. This is our Pacinian corpuscle down in those deeper reticular layers of skin. Now, as we look at the glands, we're going to cover sweat glands first, and there are two types of sweat glands that we're going to encounter in the skin. First, we're going to be looking at what are called the eccrine sweat glands, and these are exocrine in nature, meaning they're going to have a duct that goes to them. These are the most common sweat gland we have, and certainly they are going to be the ones that are controlling our elimination of salt. They're for temperature regulation to keep us cool on very hot days. And on some days, we may sweat as much as 10 liters of fluid per day. So it's really important, especially on these hot summer days here in East Texas, to stay hydrated, to drink before you go out and sweat. Now, these eccrine sweat glands are simple, coiled, tubular glands. Think of just piling up a string of spaghetti. That's what that's going to look like. And their secretion mode is going to be by merocrine excretion. Now, these sweat glands are controlled by sympathetic nerves, postganglionic sympathetic fibers. So, of course, they're going to be responding to a fight or flight situation. And the secretion sweat is very similar in its composition to what we will find as composing plasma in the blood. Here we can see an illustration from the skin. We're also illustrating a hair follicle. We'll get to those in just a bit. But here is our eccrine sweat gland, and you can see that coiled nature to the tube. This would be the duct of the sweat gland. Here we have the parenchyma of the gland itself. The duct is going to have a distinct lumen. The cells are going to be very dark, especially the nuclei. And once we look at the parenchyma, we're going to define these cell types as part of the parenchyma: the clear cell, the dark cell, and the myoepithelial cell that's going to help to express the product out of the exocrine duct and onto the surface of the skin. So in this photomicrograph, again, we have got the ducts, and we can see that very distinctive lumen. You can see how these nuclei are extremely dark. And then we also have the parenchyma of the gland. You can see some of that here, some of the parenchyma in this location. Here we have more of these parenchymal cells. It's hard to really see the lumen in these acini, but these are going to be the parenchyma where the eccrine sweat gland product is made, and they're excreted through these very pronounced ducts. Now, within the parenchyma, we're going to have dark cells, and these are the more mucus-like in their product. When we look at the base, and again, you can really only see these at the ultrastructural level. We're not going to see them at the light level. But these dark cells are basically going to be pyramidal in shape, but they're going to be somewhat upside down. So the base is going to be more toward the lumen, and the point is down toward the basement membrane. Now, it's going to be in this base where we see this glycoprotein-rich mucin that's going to be produced. And remember, once we express the mucinogen, it's going to be hydrated into mucin. And then once we add in component parts, especially debris, then we have mucus. The clear cells are going to be just the opposite. They are pyramidal shaped, but their base is against the basement membrane. They are accumulating glycogen. Now, these are going to be more serous in nature. They're going to be watery secretions, and I hate to really use serous because we think of serous as enzymatic, and these really aren't enzymes here. This is just more of the watery aqueous secretion versus the sticky, thick mucus type secretion. But here the secretion is going to be transported to the lumen through intercellular canaliculi, or enfoldings of the plasma membrane, increasing that surface area to dump as much material out of that narrow point or the apex of these cells as much as possible. Now lastly, myoepithelial cells. These are cells that look epithelial in nature because they're part of the parenchyma. However, they have contractile properties, actin and myosin. So they also have properties that resemble smooth muscle. So that's the name myoepithelial. Remember, these are around the outside of the acinus, and when these contract, it's almost like squeezing a sponge. That's going to help to express the secretory material into the lumen of the acinus, and that expression is also going to help to force the material into and out of the excretory duct of the gland. Now, the duct of the gland, typical for our sweat glands, are going to be stratified cuboidal epithelium. We're going to have a basal and luminal layer. And certainly the larger the diameter, the larger the lumen, the larger we're going to see these, and the more columnar in shape these may actually appear. But what we're going to see is that these ductal cells aren't simply transporting these materials. Ductal cells can actually remove material from the excretory product, and we'll see that these ductal cells remove potassium, sodium, chloride, and they're going to reabsorb those back into the body. But additionally, they can also secrete. Remember, we see excrete here. But when you secrete, you're putting material back in, and so you can secrete ions, urea, lactic acid, and some drugs can actually appear in your sweat on the surface of your skin. So here we see somewhat of the stratified cuboidal nature of our sweat glands. We can see one layer, an inner layer. Here we can see a smaller lumen. This is kind of tangential section, but there we see a small lumen. We have one layer on the outside, one layer on the inside, and one layer on the outside. Now this is how we know that in thick skin there are sweat glands, because this trail that we see through the stratum corneum, this is made by the duct of a sweat gland. We can't really pick it up down here in the epidermis, deeper epidermis in the dermis, but that is exactly what we're seeing there. Now the next type of sweat gland we have are not as commonly occurring throughout the body, but these are called the apocrine sweat glands. You find them in the axillary region under the armpits, around the areola of the nipple, and in the anal region, and these are more for the production of a specific odor. Some think that it's a pheromone type producing odor. However, this is the odor producing one. So when you have BO, that's where this is coming from your apocrine sweat glands. Now, these ducts do not open on the surface of the skin, but they open directly into the hair follicles. And when we look at this glandular epithelium, they're going to be a little bit taller than we see with the eccrine, cuboid to low columnar. And their type of secretion, hence the name apocrine, is the apocrine type. And these are also sympathetically controlled. So here we can see a much larger, much more open luminal diameter. Here we can see the multiple stratified. We've got the basal cells. Here we have more of these columnar cells. And we can see these down in the dermis of our skin. Now, initially, the product is odorless. However, on the surface of the skin, within the hair follicle, we have bacteria, this local fauna, and that metabolism is going to give its distinctive odor. Now, these, again, are thought to be regulated much like pheromones or these sexually attractant chemicals, and the secretion is controlled by hormones, and so there's going to be a cyclical sort of modification to when these are expressed, the odor that's present, the amount that's expressed. And so we can see these changes, especially in females, are going to be related to different stages of the menstrual cycle. Now, sebaceous glands are present not for any thermal regulation, not for the production of odor, but they are present to help condition the skin and condition the hair follicles. And so sebaceous glands are located throughout the body except where we have our thick skin. We're going to find them most abundantly on the face, the scalp, and the forehead. And unfortunately, during puberty, that's where we really have problems with acne, and that's because of infected sebaceous glands. And so the function is really to maintain this proper skin, not necessarily texture so much, but it's called suppleness, or that softness and the smoothness of our skin. And this is going to improve the flexibility of our hair. And so sebaceous glands, the product is referred to as sebum. It is a wax-like oily substance that has cholesterol, triglycerides, cellular debris, because this is going to be the holocrine type of secretion from these glands. The whole cell is going to break down and empty into the hair follicle and yield these products that are going to cause your hair to be softer, your skin to be softer. Now, the ducts of our sebaceous glands, which might be difficult at best to see, but these are going to be stratified squamous epithelium. And as I've mentioned, they open directly into the hair follicles. And when we look at the parenchyma of the sebaceous gland, it's going to be very lobular. There's going to be a number of acini, and they're very much going to resemble adipose tissue and really your multilocular adipose tissue. Let's look on the right first. This is a scanning electron micrograph of some sebaceous glands that we see here. We've cut through them, and you see how they're very honeycombed in nature. Here we have the shaft of a hair, so our hair follicles. Here's a third here. These are the sebaceous glands, and in the light microscope we can see the lobular appearance of these here. Again, looks very much like what we might expect to see with multilocular adipose tissue. But anytime you see this material associated with a hair follicle, and this is what we're looking at right here. We've even got a little slice through an erector pili muscle. It's nice they put that AP there. This is going to let us know that we are looking at sebaceous gland. Now, that leads us right into these extreme modifications of our epidermis. And so the first that we're going to look at is our hair. And basically, hair is filamentous keratin that's aligned itself very tightly together. So instead of the squams of the skin, all of these keratinized filaments of these cells have just compacted themselves together into this structure that we know as hair. And really on the human body, we have two types. We have the vellus hair and the terminal hair. The vellus hair is what we are most familiar with on our body. this very soft, fine, short, pale hair. You might think of them like your eyelashes or the hair that's present on your arm. But then we also have terminal hair, this very hard, coarse, pigmented type hair. And we see this on the scalp, on our eyebrows. That's sort of a different type of structure. But really for humans, hair is more for tactile sensation. You can really tell when someone is touching you because of the movement of the hair. Not so much for insulation in humans anymore. We just don't have the volume of hair largely that's going to help us do that like we see in some of our other mammalian relatives. So hair follicles are where hair is produced. And as I mentioned earlier, these are going to be invaginations of the epidermis. And so you think of taking a section of thick skin and just poking your finger down into this hole so that the stratum corneum is going to become the hair. And then the other different layers we have of our epidermis are going to become these layers that we can recognize of the hair follicle. And so starting from the outside in the dermis, when we look at our hair follicle, we're going to have this dense, irregular connective tissue. And then we're going to get to the basement membrane of the epidermis itself. But in a hair follicle, we don't call it the basement membrane. We call it the glassy membrane. And so now looking at what we ignored before, we looked at our sweat gland before. Now looking at our hair follicle, we're going to see that at different levels up near the surface, here in the very center, and down by the hair bulb, the actual follicle itself, we can recognize some of these layers. And in the very center, if we take this in cross section, this is what we're going to be looking at. Here we see the hair itself. And then starting on the outside, this is the basement membrane of our epidermal layer that's been invaginated, and that is our glassy membrane. From this, we will then go through and characterize these different layers. We've got the external root sheath. We then have the internal root sheath that will divide into these three layers. And then we have the actual hair itself that we'll talk about. So starting at the very base, this is where we are going to produce more and more of the cells, the hair root. And so when we look at the hair root, this is where we have living dividing cells, and they're going to be fed by capillaries. So you're going to have an invagination of the dermis into the bulb of the hair, and this is called the dermal papillae. Remember our dermal and epidermal papillae we had from our Reedy apparatus in the skin? Well, this is going to be a singular one of those injecting into the base of the hair follicle, providing a place for the capillaries to go. Now, when we look at the hair is going to have these centralized matrix cells and these outer cortical types of cells. So the matrix cells of the hair, especially down in the bulb, that's going to be the stratum basale of the skin. The central cells of the shaft of the hair are medullary. The center cells are what we think of as the center to the outer part. That's going to be the cortex, just like we saw in many of our organs. But the very outermost layer of the hair shaft itself is going to be called the cuticle. And this is going to be just adjacent to the cuticle layer of the internal root sheath. And in fact, they're interdigitating plates from the cuticle of the hair and the cuticle of the internal root sheath that are actually pointing in such a way to lock the hair in place if the hair tries to be pulled out. We'll talk about that a little bit more as we see some illustrations, and especially when we get to lab. So here we have a section through our thin skin. We can see our epidermis on the right. We're getting into hypodermis and our deeper layers here. We've got a blood vessel coming through here. We've got some muscle coursing through adipose tissue, maybe a little nerve. We've got some sweat glands here, sebaceous glands, and here is our hair follicle. We can see the shaft of the hair. Here we have the bulb, and there we have the dermal papillae projecting up into the base of the hair itself. Now, as I mentioned before, that outer layer of the hair shaft is the cuticle layer. And the next layer, as we're moving laterally now out into the epidermal type layers, is going to be the cuticle of the internal root sheath. Now imagine the cuticle of the hair has plates that are angled upward toward the surface. The internal root sheath now has plates of squams and cells that are angled pointing downward. So as the hair tries, if something tries to pull the hair out, the plate from the cuticle of the hair shaft is going to lock itself into the plate of the cuticle of the internal root sheath. And that's why you're hard pressed to pull out a hair without pulling the follicle out as well. You can't simply pull the shaft out. You know, it's that little bulb that you see on the end of the hair, because you're actually locking the hair into the follicle itself. So as we start with the internal root sheath, we've got the cuticle adjacent to the shaft of the hair. Then we're going to have two layers. The middle layer is Huxley's layer, 1 to 2 layers of cells. They're somewhat flattened in nature. Henle's layer is the most lateral layer. Now these are going to be the most challenging to see as we go through and look in them, especially depending on where you look. These are not going to be present in the bulb, so you need to look further up the shaft of the hair to really be able to see Huxley's and Henle's layer. So here we have the bulb of the hair toward the left, the shaft moving in this way. This is going to be the medulla and the cortex of the hair. And here you can see somewhat of the plates that we're talking about. You're not going to be able to distinguish which of these cuticle plates are from the shaft of the hair, which is from the follicle itself, because you see how they're at this angle. That's how they lock together, because some of these are attached to the shaft, some of these are attached to the internal root sheath, and so as you try to slide them up, they just lock into place. So this is going to be that cuticle layer, part of our internal root sheath. This is external root sheath, and there is the glassy membrane on the outside. On this side, glassy membrane, that's a basement membrane. Here we have external root sheath, and we've got our internal root sheath, including our cuticle layer. See, they don't distinguish the cuticle layer here from the internal and the hair shaft. The external root sheath, this is going to be those outer layers of the hair follicle. Again, these are those epidermal layers, the basal and the stratum spinosum. These are going to be just within the glassy membrane, and you can recognize all of those as part of the hair follicle. Once again, we can see our dermal papillae here in the hair bulb. We're not going to see, you can see how the internal root sheath and the external root sheath really disappear as we get down into the hair bulb. So we won't identify those as we get into these lower layers. You can see glassy membrane kind of slides on around here as basement membrane. Now, our last modification that we see for our integument are going to be our nails. And again, these are keratinized structures. Much like the horn of the rhinoceros is made simply of keratin, it's very different than our hair. Our nails have very different characteristics. And so the nail is going to be extremely compacted keratinized squams that are just placed so tightly together that it makes a very, very tough, strong material. Our nail plate, this is the actual stuff that we know as the fingernail, is going to grow at a rate about 0.5 millimeter a week. So it's quite adept at growing. I don't know how many times you have to cut your nails, but depending on your diet, depending on hormones, your nails may grow faster or slower. So that's the nail plate. Now, underlying the actual hard nail plate, you have the nail bed, and this is those epidermal layers underneath. So think of the nail plate, again, as stratum corneum. The nail bed, all of the other living layers of the epidermis are going to be beneath that nail plate. Now, as we look at the base of the nail plate, where we begin to have skin again, we're going to see that there is a proximal nail fold. This we often look at that as where our cuticle is. And it's going to be underneath that proximal nail fold is where we find the nail root. This is going to be the regenerative part of the nail itself. So here in our illustration, we're looking at a cross-sectional view. We have the nail bed, which they refer to as body here. This is the nail bed. Nail plate. The nail bed is going to be our epidermal layer that we see here in purple. This is going to be our dermis we see in yellow. And as we get toward this more proximal part, this is the distal part of the finger. As we get to the proximal part, here is the nail root. This is where we have the matrix that's going to begin to produce this very highly compacted stratified layer as it's pushing forward. Now, this is going to be the proximal nail fold. And as we override the nail, the root of the nail and our nail plate, we're going to have the cuticle that's going to be hanging over. Now, the cuticle is referred to as the eponychium. So that's a big fancy name for cuticle, is the eponychium, and we can see that illustrated here. Now, as we go to the distal end of our nail plate, we can see we have this region underneath the plate where it's not contacted with the nail bed. This is where you get the dirt and the debris you have to clean out. This is called the hyponychium. So below and epi above, nychium meaning the nail. That's how we can separate those two. Now we also see a little white structure here, and it's somewhat looks like a rising moon. Well, that's been referred to as the lunula part of your nail. So again, here we have in the proximal nail fold region the cuticle, which is the eponychium. That's just the stratum corneum that is overhanging that part of the nail plate. On the lateral borders, we have the lateral nail folds, and we have those grooves as well. We don't really know a function, or at least I've not been able to find one for the lunula. It's just simply that white crescent moon-like shaped structure there at the proximal nail fold that's part of the nail plate itself. And then the hyponychium is that space underneath the distal end of the nail plate. Here we have a cross section through a fingertip. Though it's maybe a finger or toe. I'm not exactly sure which. I think it's a finger. Here we can see the nail plate. We've got the nail bed. There's our eponychium, the root of the nail, hyponychium. We have bone in this location. And then here we can see our epidermis and dermis. And these ridges are going to be our dermatoglyphs. Those are our fingerprints. And you can see how those dermatoglyph ridges are mimicked with our dermal and epidermal papillae down here in the reedy apparatus layer. Again, a little bit of higher magnification. We've turned the picture around now. The distal part of the nail is to the left. So this is the root of the nail. Here we have the beginning of the nail plate. Here we have the nail bed. And then here, the stratum corneum that's overhanging, that's the cuticle or the eponychium.